Hey there, in this video we're going to be talking about how to make slides, wet mount slides for the animal cheek cell and for the elodea leaf. So there are two different methods. Both of these are wet mounts, meaning that they are slides that we have to put a little bit of water on and a cover slip to make and to use under the microscope. So first we're going to start off with a slide, and I'm going to use the pipetter or the pipette and put a few drops or a small puddle on the slide. There's a certain amount, and the amount that you want to shoot for is when you put this cover slip on, you're going to want to be able to move some water underneath, and I'll explain what that means in a little bit. For the cheek cell, what we're going to do is take a toothpick. You're going to scrape the inside of your cheek like this, and you're going to get those skin cells or these epithelial cells from your cheek, and you're going to put it right in that little puddle of water. Throw this out right away, and you're going to put your cover slip on just like this. Please make sure that when you're done with the cover slips that the cover slips do not end up in the sink, clogging the water. Please don't throw them away. Dry them off, put them back where you got them from. Same with the slides. Then, for the first part of the lab, we're going to be looking at this under the microscope. Then, I'm going to have you guys add stain. So this is iodine solution. We've used this in the past, Lugol solution. And what you do is you put a little bit, maybe like a quarter of a dropper full on one side. And as you can see, you may not be able to see this. It's only going to fill up one side. It's going to fill up one side. What you need to do is you need to drag that iodine across the slide. Pull it, pull it, pull it with capillary action. You can see I put a little piece of paper towel, and it's going to use capillary action to pull the iodine through. This is why you want to add enough water that you can allow for water movement underneath, but not so much that you have puddles of water coming off of the slide and going into the microscope. We use this method to try and eliminate air bubbles under the cover slip. They do happen, and they're pretty apparent. They look like big black dots. And so here is your wet mount of cheek cells. Next, what you would do after you're done with that is you would wash that slide, and you would use that slide again for the next part. We are going to make a slide with an elodea leaf, which is just a little bit different. It's actually just a little bit easier. We're going to put a little bit of water on the slide because it's a wet mount slide. Then we're going to use our forceps, and you want to grab an elodea leaf as close to the tip as you can, because the closer you are, the more plasmic streaming you might see. Plasmic streaming is the moving of the chloroplasts around the inside of the cell. It's really cool. And so then you just put a cover slip on, and then you have a little leaf inside. I don't know if you can see that. A little leaf inside with some water. Don't push on it. You don't want to push on anything, any cover slips, unless you're trying to crush the specimen, and we're not trying to do that. When you're creating your pre-lab, it says to draw spaces for your field of view. If we were just looking straight into the microscope, I would have you guys draw a circle, because, let's see if you can see that better. I would have you guys draw a circle because when you look into a microscope, you see a circle image. However, we are using the digital imagers, so our field of view ends up being square. So when you draw your field of view, draw those squares, and then make sure you make a list of the things that you're going to be labeling in that section. It'll make your lab go a little bit more smoothly. And if you have any questions, please ask before we get started in the lab. All right.